whether in gardens or wild corners of the earth, flowers lend an indescribable charm to the surroundings in which they grow. Widely used as objects of ornamental, social, religious, and cultural value, flowers also are sites of sexual reproduction. Flowering is a major phase in the life cycle of a plant and takes place when the climate is congenial for pollination and the formation of seeds. This phase is, however, preceded by several physiological and morphological changes induced by hormones such as florigen present in the leaves. These changes lead to the differentiation of vegetative buds into floral buds and the development of the floral primordium, the rudimentary or preliminary stage of the flower or the flowering shoot. This primordium either bears a solitary floral bud or inflorescences that bear multiple floral buds. The buds bloom into a flower, a modified shoot with shortened internodes and nodes. From these nodes arise four modified leaf-like structures called whorls, which are arranged on the swollen end of a stalk called the thalamus. The whorls include the androecium, gynoecium, calyx that consists of sepals, and lastly the corolla comprising the petals. The calyx and corolla, referred to as the perianth, are bright and colorful to attract insect pollinators. The gynoecium, on the other hand, which consists of one or many carpels or pistils, represents the female reproductive part, whereas the androecium, which consists of a whorl of stamens, is the male reproductive part of the flower. Though the number and size of stamens in an androecium differ across species of flowers, the basic structure remains the same. Every stamen, for instance, consists of a filament and an anther. The filament is a long and slender stalk whose proximal end is attached either to the thalamus or the petal of the flower. The anther, on the other hand, is bilobed in nature, with each lobe being dithecous or consisting of two thecas that are separated by a longitudinal groove that runs through the anther. The transverse section of the anther reveals that its lobes form a four-sided or a tetragonal structure. In the corner of each lobe is the microsporangium, a sac-like structure which is nearly circular in outline. In a young anther, the microsporangium is surrounded by a multi-layered wall consisting of the epidermis, endothecium, middle layers, and the tapetum, the innermost layer, while the center is occupied by sporogenous tissue. The epidermis, endothecium, and middle layers protect the microsporangium as well as aid in dehiscence, which is the splitting of the anther walls that causes the release of pollen grains. While the tapetum, whose binucleate cells possess dense cytoplasm, nourishes the developing pollen grains, the sporogenous tissue, on the other hand, consists of a group of compactly arranged homogeneous diploid cells called microspore mother cells or pollen mother cells. These microspore mother cells divide meiotically to form haploid microspores, which arrange themselves in a tetrad. The process of formation of microspores meiotically from a microspore mother cell is called microsporogenesis. As the anther begins to mature, both the microsporangium and microspores undergo changes. While the microsporangium develops into the pollen sacs that extend longitudinally through the length of the anther, the microspores separate from the tetrad and develop into pollen grains. Thus, the anther contains thousands of pollen grains which undergo mitosis to form two unequal cells. The larger cell, known as the vegetative cell, is characterized by a large, irregular-shaped nucleus, vacuoles, and abundant food reserves. While the smaller cell, known as the generative cell, is distinctly spindle-shaped and possesses dense cytoplasm. Moreover, the generative cell floats in the cytoplasm of the vegetative cell. Did you know that the vegetative cell and generative cell represent the two-celled stage of the pollen grain? Later, the generative cell undergoes mitotic division to produce two male gametes. The pollen grain is now said to be at the three-celled stage. After the formation of pollen grains, the endothecial cells lose water, which leads to tension in their cell walls. This tension causes the dehiscence or bursting of the anther along the line of dehiscence and results in the shedding of several thousand pollen grains. Are you aware that in cereals such as jowar, the pollen grains are shed at the three-celled stage, whereas in 60% of angiosperms, which include plants such as hibiscus, the pollen grains are shed at the two-celled stage? Pollen grains, which represent the male gametophyte, come in a wide variety of sizes, shapes, and colors. A typical pollen grain, however, is spherical in shape and has a diameter measuring about 25 to 50 micrometers. It is surrounded by two layers of wall, exine and intine. Exine, the hard outer layer, is made up of sporopollenin, a tough organic material capable of resisting high temperatures and strong acids, alkalis, and all natural enzymes. Hence, pollen can be well preserved as fossils. However, sporopollenin is not present in the germ spore, an aperture present in the exine through which we see the germination of the pollen tube. Intine, on the other hand, is a thin and continuous inner layer of wall composed of cellulose and pectin. On the inner side of the intine is the plasma membrane that surrounds the pollen grain's cytoplasm. When pollen grains land on the stigma, they germinate into a pollen tube that carries the male gametes towards the embryo sac. This ability of the pollen grain to deliver the male gametes to the embryo sac is called pollen viability. However, factors such as temperature and humidity affect pollen viability. Moreover, pollen grains of different plants have different periods of viability. Pollen grains of cereals such as wheat and rice lose their viability within 30 minutes of their release. Whereas pollen grains of some members of Rosaceae, Leguminosae and Solanaceae remain viable for months. Pollen grains, which play a vital role in plant reproduction, are also often consumed by human beings as they are rich in nutrients. Pollen products, in the form of tablets and syrups, are commonly spotted on shelves of supermarkets. Moreover, pollen is also stored in pollen banks in liquid nitrogen at 196 degrees centigrade. The pollen is later used in plant breeding programs. Pollen grains, however, can trigger allergies in some people and can cause asthma and bronchitis. In fact, the weed carrot grass or Parthenium hysterophorus and its pollen, found in non-cultivated lands in Punjab, causes allergies such as eczema, dermatitis, and other skin diseases. Pollen, formed inside the stamen, plays an important role in plant propagation and also serves as a source of nutrition, although in some cases it may trigger allergies. Flowers are one of the most fascinating sights to behold. Incidentally, flowers also happen to be the site of sexual reproduction, as they contain the androecium and gynoecium, the male and female reproductive organs of a plant. You can see many variations in the gynoecium of different flowers. For example, the gynoecium can be monocarpous, consisting of a single pistil or carpel. Or it may be multicarpous, in which case we see several pistils in a single flower. Moreover, a multicarpellary gynoecium may be apocarpous, that is, the pistils are free, or syncarpous, where the pistils are fused. If you observe the structure of a pistil, you will find that it consists of the stigma at the tip, followed by the style in the middle and an ovary at the base. While the stigma is a landing platform for pollen grains, the style, an elongated, slender structure, connects the stigma to the ovary, the basal bulging part of the pistil. Morphological studies of the ovary have revealed that it contains one or more cavities called locules, or ovarian cavities that are surrounded by an ovary wall. Within the locule, you will find ovules or megasporangia that are attached to the ovary wall in a region called the placenta. These ovules may be arranged in free central, axile, marginal, parietal, or basal placentation. Moreover, after fertilization, the ovules form seeds, while the ovary develops into a fruit. Did you know that different flowers of different plant species have a varying number of ovules in their ovaries? A peach, for instance, develops from an ovary containing a solitary ovule. A papaya, on the other hand, has several ovules in its ovary, and that's why you see numerous seeds in the fruit when it is split open. The ovule, considered by botanists as the forerunner of the seed, has an oval or egg-shaped body. It remains attached to the placenta by a stalk called the funicle. In fact, the ovule's body fuses with the funicle in a region called the hilum. The ovule is well protected with one or two protective coverings called integuments. These integuments later form the seed coat. The integuments cover the entire ovule except at the apex, where they form a narrow opening called the micropyle. Did you know that the micropylar end acts as an entry point for the pollen tube during the process of fertilization? While the micropyle forms one end of the ovule, the chalaza forms the basal part of the ovule. At the chalazel region, we find that the funicle and integuments join the nucellus, the center region of the ovule that's made of a mass of diploid, colorless, thin-walled, parenchymatous cells containing food reserve materials. In the nucellus of a mature ovule lies the embryo sac, the female gametophyte, which develops from a megaspore. This megaspore is formed when one of the nucellus cells, towards its micropylar end, gets differentiated into a megaspore mother cell, which can be easily distinguished from other cells due to its large size, dense cytoplasm, and prominent nucleus. The megaspore mother cell undergoes meiosis to form 4 megaspores which arrange themselves in a linear tetrad. This process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Of the 4 megaspores, usually the one near the chalazel end becomes functional, whereas the other 3 near the micropylar end degenerate. The functional megaspore enlarges and simultaneously undergoes mitotic division to form an embryo sac. This type of formation of the embryo sac from a single megaspore is known as monosporic development. While undergoing these mitotic divisions, the nucleus of the functional megaspore first divides to form 2 nuclei, which move to the 2 opposite ends of the embryo sac. This is the 2-nucleate stage of the embryo sac. The mitotic divisions continue and result in the formation of the 4-nucleate and later the 8-nucleate stages of the embryo sac. A mature embryo sac thus has 8 nuclei after 3 mitotic divisions, which are arranged in a group of 4 at each end of the embryo sac. Interestingly, the mitotic divisions that occur in the megaspore's nucleus are free nuclear, which means the division of the nuclei doesn't immediately trigger cytoplasmic division and cell wall formation. Instead, we see the formation of cell walls only after the 8-nucleate stage, due to which 6 of the 8 nuclei organize into cells. The 2 remaining nuclei, called polar nuclei, migrate to the center of the embryo sac. Thus, we find the embryo sac to be in 7-celled and 8-nucleate stage with 6 cells at the poles and a large central cell with 2 nuclei in the center. Meanwhile, the 6 cells organize to form the typical structure of the embryo sac, which is now ready for fertilization. The 3 cells present at the chalazial end, for instance, group together to form the antipodal cells, which do not have any specific function, whereas the 3 cells at the micropylar end group together to form the egg apparatus. While one of the cells functions as an egg or the female gamete, the other 2 cells are called synergids. If you observe the micropylar tip of the synergids, you will notice special cellular thickenings called a filiform apparatus that guides the pollen grains into the synergid. The synergids, as well as other cells, form the embryo sac, the female gametophyte born inside the pistil. Flowers are mainly responsible for bringing about fertilization and seed formation. To facilitate fertilization, it is necessary that the male gametes inside the pollen grains and the female gamete inside the ovule are brought together. However, since the gametes are immotile, pollen grains have to be transferred to the stigma. This process of transfer of pollen to the stigma of a flower, brought about by agents such as insects, wind and water, is called pollination. Pollination can be of 2 types, self-pollination and cross-pollination. When pollination takes place within the same flower or between flowers of the same plant, it is known as self-pollination. In plants, self-pollination can occur either through autogamy or geitonogamy. In autogamy, pollen is transferred from the anther to the stigma of the same flower. And is seen in cleistogamous flowers or flowers which do not open at all. Produced by plants such as viola, oxalis, camelina, and Arachis hypogea or peanut. The anthers and stigma of these flowers lie very close to each other. When anthers dehisce, pollen grains get deposited on the stigma, which ensures the production of seed sets even in the absence of pollinators. Autogamy, however, is rare in chasmogamous flowers, which open and expose their anthers and stigma, as it requires synchrony between pollen release and stigma receptivity. In geitonogamy, the second type of self-pollination, pollen is transferred from the anther of one flower to the stigma of another flower of the same plant. Apart from self-pollination, cross-pollination is also commonly seen in plants. This type of pollination involves the transfer of pollen grains from the flower of one plant to the stigma of the flower of another plant of the same type. This type of pollination is called xenogamy, or allogamy. Interestingly, xenogamy is the only type of pollination that brings genetically different types of pollen grains to the stigma. In both cross-pollination and self-pollination, pollinating agents play a vital role. These agents include biotic agents such as insects, birds, bees, butterflies, and mammals such as bats, as well as abiotic agents such as wind and water. Incidentally, pollination in a majority of plants is aided by biotic pollinating agents, which get attracted by flowers which vary from each other to attract their specific pollinator. Insect-pollinated flowers, for instance, are usually large and fragrant. However, if they are small, they are bunched together in an inflorescence to make them conspicuous. Moreover, these flowers produce pollen grains as well as large quantities of nectar to reward the agents. When a pollinating agent comes in contact with the anther, its body gets covered in a coat of sticky pollen grains. When the agent sits on another flower to suck the nectar, the pollen present on their bodies comes into contact with the stigma, thus completing the process of pollination. Conversely, flowers pollinated by butterflies are usually lightly scented and brightly colored, with nectar not very deeply hidden, while those pollinated by beetles and flies secrete a rotten odor. Flowers pollinated by birds, on the other hand, have large, odorless orange or red tubes which are rich in honey. Certain plants attract pollinating insects by offering them shelter. For instance, Amorphophallus, a 6-foot-tall flower, attracts pollinating insects by offering them a safe haven to lay their eggs. In the yucca plant, on the other hand, a female moth transfers the pollen of another flower to the stigma and simultaneously deposits her eggs in the locule of the flower's ovary. The larvae, which hatch out of the eggs, start consuming some of the developing seeds, while leaving enough of them to propagate the plant. Thus, both the moth and the plant are dependent on each other for the completion of their respective life cycles. However, certain birds and insects such as the bumblebee visit flowers for nectar and pollen, but don't bring about pollination. These creatures are referred to as pollen robbers. Honeybees, as well as other insects, also pollinate a large number of aquatic flowering plants, such as the freshwater lily. However, in certain freshwater plants such as Vallisneria and Hydrilla, and marine seagrasses such as